All right, looking at the generalized cell, this is a typical picture in any textbook, especially biology books, obviously. And this is what we would call a generalized or typical animal cell. In the human body, there are really no cells that look like this. This is just an artist's attempt at getting as many organelles crammed into one picture as possible. This is actually from a different textbook than your current textbook, so there are some leader lines that are in a little different location than the ones that you might have in your book. But generally speaking, everything's the same. There are a few leader lines on here that are a little bit ambiguous. They're kind of sticking out in the middle of nowhere, so we'll have to make a little bit of creative license in deciding what those things are pointing to. But I think for the most part, it'll make sense. There is no real place to start, so I think I'll start up here towards the top with the nucleus. And generally speaking, we're just going to label the nucleus right here, and that is this large purple structure in the center. It does have in the center of the nucleus, and there may be more than one of these depending on what the cell's job is, something called a nucleolus. And its job is to manufacture ribosomes. It's a site of intense transcription. Our RNA is manufactured there and then later assembled into ribosomes. So the nucleolus' job is to make ribosomes. This purple diffuse stuff that goes all the way around, this is called chromatin. This is DNA with associated proteins. So you'll find that in a working nucleus, you'll see a lot of this chromatin. When cells are dividing, when they're going through mitosis, or in the case of gametes going through meiosis, this chromatin will be condensed into chromosomes. I believe this leader line is pointing to one of these holes. It could be the nuclear envelope, or it could be the hole within the nuclear envelope. So there are little pores here. So we'll call this a nuclear pore, but it is a hole or an opening in the nuclear envelope, a lipid layer that's actually a double membrane layer that surrounds the nucleus and separates it from the cytoplasm. This leader line here is the plasma membrane. We looked at its structure in the previous YouTube video, so you can go back and look at that if you'd like to. This is rough ER, and I can tell it's rough ER because it's a membranous labyrinth there, kind of a maze of membranes with these little speckles on it, and the speckles are actually ribosomes. There are two kinds of ribosomes in a cell, free ribosomes and attached ribosomes. So these are attached ribosomes here because they're actually embedded in the rough ER. Over here, I'm going to go way over on the left-hand side. These are free ribosomes, a little cluster of them here. There's a few more hanging out down here. Now I'm going to get rid of those circles so they don't confuse you. But there are some subtle differences between the two ribosomes. Free ribosomes manufacture proteins that stay inside the cell for the most part, and attached ribosomes, those that are attached to rough ER, those are involved in protein synthesis, and the proteins generally leave the cell. Not always. Sometimes they're packaged inside organelles, but usually they're not floating around inside the cytosol or the cytoplasm. This is the Golgi apparatus. Its job is to package and modify proteins, especially those for export. So you can see in this picture that there are some proteins in here and here that are being modified, and here they are completed. The modification has been complete, and it will actually go through a process called exocytosis where the vacuole, a little transport vesicle in this case, will bind to the plasma membrane and dump the contents out, and that's what we're seeing here. These are contents being dumped out. So this is some sort of secretory, whoops, I spelled that wrong, some sort of secretory product. So for example, if this were a pancreas cell, a special cell in a section of the pancreas known as the islets of Langerhans, if this were a beta cell, it would be involved in producing a protein, a hormone called insulin, and so those would be insulin proteins being released into the bloodstream. I don't know if that's what they're supposed to be, but that's a good example anyway. Down here is a peroxisome. This is a unique organelle. Its job is to help detoxify. That's a little misleading because we have another detoxifying organelle as well. This one's involved in getting rid of these very, very dangerous free radicals, these superoxides that are made during parts of our metabolism. And what the peroxisomes do is they convert that bad oxide, that super free radical of oxygen, into hydrogen peroxide. Now this substance looks like water, but there's actually an extra O there, so it's H2O2, and it is highly toxic. So we have some oxidase enzymes that help produce the superoxides into peroxide, and then we have an enzyme called catalase that then decomposes hydrogen peroxide into something as safe as water along with oxygen gas. So this peroxisome is a little organelle that is loaded with enzymes and helps convert some metabolic byproducts along with some toxins. Your book does mention that alcohol and some other poisons are detoxified here. That is something new that's just being brought out. Other organelles were thought to detoxify alcohol, but we'll go ahead and just say that for right now, peroxisomes are involved in the production of hydrogen peroxide, and then that's decomposed into water and oxygen gas. The peroxisome gets its name because of the hydrogen peroxide. Next organelle around the corner is part of the cytoskeleton. These are intermediate filaments. These are the most stable of the cytoskeleton. They kind of give the cell some shape, and once they're formed, once they're laid down, they don't tend to change much. So anchoring things to the cell membrane, anchoring things to the nucleus, that's what these guys do. This leader line is out in the middle of nowhere, this guy, so I'm going to assume that this is the cytosol. Let me spell that right here. Cytoplasm or cytosol. I've been using both terms. They're not interchangeable technically. Cytosol is the fluid-like area, fluid-like substance, where cytoplasm is the area. This leader line is pointing to some centrioles. These occur in pairs. So there's a centriole here, and then there's another one kind of going down behind the scenes that you can't see very well. These centrioles basically don't do anything unless the cell has a flagellum or a cilium. If the cell has a flagellum or a cilium, or multiple cilia typically is what happens, these centrioles will become basal bodies. And basal bodies anchor cilia or flagella so that they won't detach from the plasma membrane when they're doing their job. So centrioles in cells, for instance, this cell does not have any cilia or flagella, so these centrioles are just hanging out doing nothing. They are found in the center of a structure called a centrosome, and the centrosome is more of an area here, so this is the centrosome, and from the centrosome you can see these tubules migrating out from that center. Those tubules, and I believe that's what this leader line up here is trying to identify, are called microtubules. So it used to be thought that centrioles were involved in the production of microtubules, but somebody with a creative and devious mind shot out the centrioles with a laser, and the cell was still able to produce microtubules. So the centrioles are not involved in microtubule production, although they're in the center of the centrosome, which is involved in microtubule production. Microtubules aren't as stable as intermediate filaments, so they're constantly being moved and changed, and they grow and they shrink, and they help move organelles around inside the cell. They also help chromosomes move during mitosis, so we'll talk a little more about those guys later on. Let's see, we got a couple left here. This one is a mitochondrion. There are two mitochondria, actually three. There's one up here in the corner. These are energy production organelles. Their job is to manufacture ATP through a process called aerobic respiration. They do require oxygen to do their job. They have this very elaborate membranous system on the inside called cristae, and the cristae actually give them more surface area so they can have more electron transport chains and areas for various enzymes to be embedded. So that's what that little organelle is all about. The last organelle that's kind of hiding under there is a lysosome. And a lysosome is a very important organelle that's involved in the degrading or digestion of things that the cell needs to get rid of. If this were a white blood cell and it was phagocytizing and eating bacteria and damaged cells, the lysosome would be full of hydrolytic enzymes that would allow the white blood cell to digest that bacteria or damaged cell. So you can think of it as the cell's stomach. That's what many sophomore biology teachers use as an analogy. We're going to learn later on that the stomach really isn't a site of a lot of digestion. Most of the digestion happens in the small intestine. So I guess a better analogy would be it's the cell's small intestine, but that's just kind of being weird. I thought that was the last one, but it's not. We do have smooth ER up here at the top, smooth endoplasmic reticulum. Smooth endoplasmic reticulum has three jobs. It is the site of lipid metabolism. So lipids are manufactured there along with not only being made, but also that's where some lipids are metabolized in that spot. They're actually broken down in that spot or catabolized. It's also a site of detoxifying. So older textbooks claim that alcohol and other toxins that you might consume, for instance, if you ate a mushroom in the forest that was actually poisonous, it would be the job of the smooth ER of your liver cells to decompose and get rid of that toxin if it could. Sometimes it's not able to do that, and then obviously you die of the poison. Another thing that the smooth ER is famous for is storage. it can act as a calcium storehouse. So calcium, specifically calcium ions, in muscle cells are going to be stored in that smooth ER. Whoops, I didn't spell smooth right. There you go. Smooth ER in muscle cells has a different name. We'll call it by its different name later on, but for the testing situation, you'll want to know that smooth ER can act as a storehouse. So in case I lost you somewhere in there, it has three jobs. It's involved in lipid metabolism, either the manufacturing and or breakdown of lipids. It's involved in the storage of calcium, and it's involved in detoxifying. For our purposes, the smooth ER is going to be detoxifying, and the peroxisomes are going to be converting free radicals into hydrogen peroxide. That'll help us keep track of them. But a liver cell that's involved in detoxifying things would have a good supply of both organelles. One thing that's kind of interesting about smooth ER is that very few cells have smooth ER. It's really concentrated in cells that are involved in making lipids or processing lipids or detoxifying. So your liver cells are going to have a lot of smooth ER. Testes, the testicles, they are involved in making a steroid, which is a type of lipid. So testes would be testes cells. Special cells called Leydig cells would be loaded with smooth ER so they could manufacture and release testosterone. And then, of course, those skeletal muscle cells for sure have tons of smooth ER where they store a bunch of calcium. All right, so let's see. I think I gave you the job of most of these as I went around. So just in case I forgot one, I don't remember if we talked about rough ER. I know we talked about ribosomes and protein synthesis, but rough ER would be involved in protein synthesis. We mentioned the membrane, so I think that takes care of general organelles. Okay, I think I'm going to do one more thing with this section. I think what I'm going to do is skip through this and just go down to this picture right here. This is showing us some of the different kinds of cell junctions and cell processes that cells have. Let me zoom in on that a little bit there, make it a little bit bigger. So we've got some cells here. It looks like we've got one cell, two cells, three cells, and then a piece of a fourth cell here. And there actually might be another piece of a fifth cell here. So we've got 5 cells that are all sitting side by side. We'll soon learn that these are epithelial cells, and specifically these are called simple columnar epithelial cells. So these would be some cells that possibly could be lining your intestine. Well, we have to have ways of these cells interacting and hanging on to one another. So the first thing we have is that we have a space or a gap in between the cells. There is a little space or gap in between all of these cells, and that's an intercellular space or an intercellular gap. So the cells are held close together, but there is still some space there, and there'd be some interstitial fluid that could flow through. Now, if this is your intestine, this would be called the lumen of the intestine. That's where the food would be. So any food that you're eating, that you're digesting, is going to be in this section. So if we're going to absorb any food, it's got to go down this way into these cells. What we don't want to have happen is we don't want the food to be able to squeeze through these cells unprocessed and get down into our bloodstream. So we have to have a way of preventing things from leaking through. We need to have a way of making this an impermeable barrier. And that's what this section right here is all about. Now let me get rid of some of these scribbles so it's not hard to see here. But that section right there, these special little lines there are called tight junctions. Tight junctions. And they form a waterproof impermeable barrier. So I guess we're zooming in on them over here. So these are the tight junctions right here. And this is just pointing to, right here, that's just pointing to the intercellular space. So you can see that we've closed off the intercellular space, so nothing can sneak through. It's going to get actually blocked from passing through. It's not a terribly strong junction, but it's definitely waterproof and impermeable. There are a few exceptions to that, but for the most part, that is a true statement. Okay, the next kind of junction we have is with the square here, and you can see that we're zooming in down here at the bottom. This is called a desmosome. So the desmosome, let me write that down here for you, desmosome. The desmosome is an anchoring junction. It holds cells together. What it's actually doing is joining the cytoskeletons of one cell to the cytoskeleton of another cell. So we've got these long intermediate filaments here. These are part of the cytoskeleton, and these are being linked together here by these little proteins here that are going to adhere them together. These are called cadherins, and they hold them together, so it's nice and tight. So this desmosome will hold these guys together so that it's strong. They won't separate it. It'll keep that tissue from tearing or coming apart. It'll keep these cells from being dislodged from one another. There are many desmosomes in your skin, and that's why your skin doesn't fall off your body when you pull on it. Okay, the final junction is this guy right here. These are called gap junctions. These are communicating junctions. So this allows the cells to communicate. They can pass chemicals to and forth back through this little channel. These little channels are made by little small structures called connexons, and so these connexons connect the two cells together so that we can send, or so that the cells can send chemical messages back and forth. So those are the main three types of junctions. Tight junctions, which are watertight or impermeable barriers, desmosomes, which are anchoring junctions, and gap junctions, which are communicating junctions. Now the only other thing that's showing up on this diagram is that cells that do absorption, so there's cells here that do absorption, have extensions of their plasma membrane here that increase surface area, and these are called microvilli. It's actually one word. I've got it looking like it's two, but it's actually one word, microvilli. And that increases the surface area so that the cell can actually absorb, in this case, nutrients. So if food is going to be absorbed into your bloodstream, it's got to go through the cell so it can be processed before it's passed on. We don't want it to sneak through unprocessed, so we have to have it processed. We want the cell to have a chance to process it and distribute nutrients to the right spot. We'll talk a lot more about that in the digestive system chapter, but for right now, microvilli, those are extensions. Do not confuse microvilli with cilia. Cilia and microvilli are not the same. These are extensions of the plasma membrane. Cilia are actually hair-like structures that actually sweep back and forth, and typically they're involved in creating currents or moving mucus, and we'll look more at that in chapter 4 when we do some tissues. We'll have some ciliated tissues, and you'll see those. I can also tell that these aren't cilia because there's no basal body anchoring them. There had to be a basal body down here anchoring each one of those, and there isn't. That's just simply a way of increasing surface area for absorption. All right, so that gets us through that. The rest of this stuff here is dealing with how we transport things across membranes, and we'll talk about that in the next discussion. So that should take care of it for right now, and I'll catch you some other time.